Cancer, in general, is characterized as a group of heterogeneous cells, growing rapidly in an uncontrolled and disorganized fashion. During the multi-step process of tumorigenesis, the tumor increases in size to produce a large mass. Many treatments have been developed to kill cancer cells. Most of these treatments work by targeting rapidly proliferating cells in general. The typical result is reduction in tumor bulk and slowed growth. In spite of the recent advances in chemotherapy, recurrence and metastasis remain challenges in clinical practice. Emerging preclinical evidence suggests that there are subpopulations of cancer cells which do not proliferate rapidly, but are nonetheless responsible for tumor recurrence and metastasis, and are resistant to standard treatments. These relatively quiescent cells are called cancer stem cells. Cancer stem cells interrelate with their tumor microenvironment niche to maintain their stem cell-like properties via interactions with the extracellular matrix and activation of various signal transduction pathways. These cancer stem cells have the capability of self-renewal and give rise to a variety of proliferating and differentiated cells that comprise the majority of the tumor bulk. The presence of cancer stem cells has been reported in many types of cancer. Furthermore, recent reports have suggested that these cells are far more tumorigenic than non-stem cancer cells. Activation of these cell signaling pathways can promote epithelial mesenchymal transition, which enables cancer cells to become mobile and intravasate into lymphatic and blood vessels. The cells are then transported via the general circulation to distant organ sites as seeds of metastasis. The tumor microenvironment in the metastatic sites maintains their stem cell-like properties and also promotes drug resistance through the interplay of cell adhesion and cell signaling molecules. As cancer stem cells are able to evade conventional chemotherapy and radiation treatments, when such treatments are applied to a tumor, cancer stem cells may persist. Persistent cancer stem cells may cause recurrence and metastasis requiring further treatment. Focusing on the signaling pathways involved in acquisition or maintenance of stem cell-like properties has potential to yield novel therapeutic targets. Current research suggests that targeting the properties of cancer stem cells or their tumor microenvironment might be an effective new way to treat cancer. Therapies that target cancer stem cells might have the potential to overcome treatment resistance, to reduce metastasis and tumor recurrence, and to potentially improve clinical outcomes for patients with cancer.